Welcome everyone to this month's MycoTalks. My name is Gordon Brown and I'm based here at the MRC Centre for Medical Mycology at the University of Exeter. And my co-chair today is Professor Sarah Gavin from the University of Pittsburgh. So as always, we have two fantastic speakers lined up for you today. The first will be Professor Aleeza Gerstein from the University of Manitoba, who's going to tell us about the evolution of human fungal pathogens. Followed by Professor Stuart Levitz from the University of Massachusetts, who's going to talk to us about cryptococcal vaccines. So if you haven't joined us before, just to give you just a very brief update. So an overview is that the purpose of the seminar series really is to provide the medical mycology community with an open and access platform in which we can all share in the wonderful diversity of the research that we're undertaking. Every MycoTalks is composed of two talks, each 30 minutes in length, and each talk follows the other. And it's only at the end of the two presentations that we will have questions. Now, to submit questions to the speakers, please can you use the Q&A function that you'll see at the bottom of your slide. We will not use the chat function, so the Q&A button is mandatory and is what will be required in order for us to get the questions that you wish to ask. And these questions are not shared with you, but we, Sarah and I will summarize them and then we'll tag team at the end of the talks to present the questions to the speakers. So we record all of the talks, so if you want to forward interesting points that you want to raise with other people, you can point them to our website that you can see that listed there on the page, where we store all of these talks. I just need to remind you that you need to register for every monthly MycoTalk, and we have another really good lineup coming up in March and April, as you can see. And please do use the link on our university website to be able to register for your interest in attending to these talks. And then finally, just to remind you about two other related seminar series that we have. The one is the MycoClinics, which is focused on the clinical aspects, as you can see in the slide. And the next one is in March. And then this is also supported by another series that we have for trainees, the Trainee Seminar Series, which is a really fantastic opportunity for trainees and early career researchers to present their work to a global audience. And I should also point out that every year we select one of the speakers from the series to speak in our MycoTalks series. So the best speaker gets to present at this very MycoTalk. And then with that, I will hand over to my co-chair, Sarah, who will introduce our first speaker.
Thank you, Gordon. I'm Sarah Gaffin from the University of Pittsburgh, and it's my great pleasure to introduce our first speaker, Aleeza Gerstein, who's coming to us from the University of Manitoba. Aleeza did her undergraduate work at the University of Western Ontario, working on ecology and evolution, and proceeded to do her PhD at the University of British Columbia, working on Saccharomyces genetics and beginning to develop her expertise in computational approaches to studying evolution of fungi. She did postdoctoral work at University of Minnesota and Tel Aviv University, where she moved into medical mycology, pathogens, Candida and crypto, and really has positioned herself to become a leading expert on computational approaches to evolutionary genetics. Since 2018, she's been at the University of Manitoba and has some really exciting work in this space. I can't wait to hear it, with a lot of implications, of course, for understanding not only fungal population genetics, but the implications for clinical disease, including drug resistance. So finally, just a reminder to everyone listening, please use the Q&A function for your questions, not the chat. We will be monitoring them, and please indicate to whom your question is directed. All right, with that, take it away, Aleeza.
Thank you so much. All right, let's see if I get this right on the first try. All right, no one interrupts me to say something is amiss, then I will get started. All right, so thank you so much for this invitation. It's really exciting to get the opportunity to speak to this group today. I've loved listening to many of the previous talks. So today, as Sarah said, I'm going to tell you about how evolutionary forces shape yeast populations. And we're going to look at this from a single individual to the global relationships. And this is work that has been done by my first PhD student, Abdirahman Adami Bukhari. He is approaching the end of his degree, and he unfortunately told me he's thinking about jumping back into bacteria because of how challenging fungi are. But maybe one of you can help me convince him to stay in fungal biology. I think he's done some wonderful work. Okay, so this is sort of what's driven me since I was an undergraduate student. So this is a photo from the Field Museum in Chicago with Darwin's quote in the middle. So from so simple a beginning, endless forms, most beautiful and most wonderful have been and are being evolved. And I've started many, many, many talks with this slide and this quote because this really has sort of gotten me up in the morning now for 20 years, just thinking about what is it that shapes the way that we see life around us. So when we go into an ecosystem, what has determined why some organisms are there and some are not, and how are the things that exist in that space interacting with each other in both beneficial and antagonistic ways. I started off my academic journey looking at Saccharomyces cerevisiae, and this was very much from a model organism perspective. So Saccharomyces cerevisiae is a fantastic model for studying evolution in the lab. I discovered that we could evolve them in test tubes and high replication and study all kinds of targeted questions about them. But there was something very unsatisfying about that. So I was in graduate school with people who studied fish and birds and plants in these amazing ecosystems, and they could precisely sort of take organisms from different places and study how they differed depending on the environment they found in. And so what I realized towards the end of graduate school is that I didn't have to leave microbiology and I didn't have to leave fungal biology. What I could do instead is switching from studying Saccharomyces cerevisiae to switching human fungal pathogens where we do know where they are coming from. So I don't think I need to tell you all this, but let's have a moment to appreciate how amazing fungal opportunistic pathogens are. This is a schematic of Candida albicans. As we know, it can grow as both a commensal and a pathogenic, and it can grow all throughout the body in a huge variety of places. So it can grow in the oral cavity, the nasal cavity, the skin, in the GI tract, in the vaginal tract. It grows as part of the commensal microbiome. And then something happens and we know something about what that something is. And we also have a lot of questions in some cases about what that something is, where it switches from being a commensal to a pathogenic. And instead of existing quite happily where the person probably doesn't even know they have Candida there, it can cause disease. And I think there's still a lot of very interesting questions about what that switch is to go from commensal to pathogen. So one of the things that I think is so amazing about Candida albicans, if we think about all these different sites, they vary in all kinds of abiotic ways. And so if we look at the oral tract compared to the vagina, for example, this differs in pH, they differ in temperature, it differs in oxygen tension. And so Candida is able to seemingly happily exist in all of these different places in very closely related forms, as I'll tell you about. We see the same essential genotypes living in all these different places, subjected to all these different abiotic pressures quite happily, which is really an amazing thing for an organism to accomplish. That's not true of all that many species on the planet, that they can just grow happily in all these different conditions. In addition to the abiotic pressures that they have to face in this diversity, they're also growing with all kinds of biotic organisms. So this is just sort of a high level of bacterial genera. The specific colors of the pies doesn't really matter. The takeaway is that they're really, really different. And so the species they're coexisting with in these very different environments quite happily is, I think, remarkable. But this also raises the question, which is how do you adapt to an environment? Can you adapt to an environment when you face so many different pressures? How do you deal with this? And what is the sort of predominant thing in which you are evolving towards? And so before I really get into the nuts and bolts of my talk, I want to make sure we have some common language. And so if we think about the human body as a fitness landscape. And so apologies to Moana, but I recently watched this with my kids, and as soon as I saw this scene, I thought, this is perfect. I'm going to use this the first opportunity I get. So here we are. Okay, so evolutionary biologists love metaphors, and one of our favorite is the fitness landscape. And so if you think about a mountain range as a fitness landscape, organisms are trying to climb the mountain. So they go from having a population, and then they incorporate mutations to get higher fitness. And so if we think about some little yeast population at the bottom of this mountain, or the bottom in some place in the human body, what they're trying to do is incorporate these beneficial mutations that arise in that population somewhat randomly, to incorporate them so that the beneficial mutations sort of take over the population. And so here we see 4 different populations, yellow, pink, brown, and purple, which have all acquired different mutations and evolved to be better at growing in those different places. And so this is how I think a lot of us think about how the world works. Selection acts on populations. Populations get better at the environments that they're in. But selection is not the only force that is acting. We also see in lots of scenarios, prevalent migration or gene flow. And so this is where populations between one side and another are interacting. And so the alleles that might be good at one place get moved over to another place just because things are moving around. We also see drift or stochasticity. So although I anyways tend to think about microbial populations as quite large, which is where selection is really efficient, drift is also potentially really important. So this is where we'd have population bottlenecks or population crashes due to things like taking drug. And then the dynamics that determine which mutations sort of make it through are much more based on chance than they are necessarily the things that are good. And so I just want to make sure that we're clear that just because we see changes in allele frequencies in a particular place doesn't necessarily mean that those changes are good. It doesn't necessarily mean this is a signature of adaptation. So what do we need for adaptation? So we need selection, which requires genetic diversity. So there have to be differences for the population to act on, for selection to act on. And adaptation critically requires selection to overcome drift and migration. So if we ask the question, are yeast populations adapting? We certainly have some great examples of this happening in the human body. So this remains one of my favorite studies. This is from the Ford et al. Paper published in 2015. And this is a Ted White series of isolates. And so these are 14 isolates taken from the same person. So we're starting at day 0 up to day about 700. The person took ever-increasing dosages of fluconazole, shown here in different colors of pink. And as time went on and the dose increased, the MIC, or the resistance level of that time series of isolates, also increased over time. And so this is what we expect as like a clear response to selection. Population was exposed to a selective pressure drug, and it got better at dealing with that selective pressure, unfortunately for the person involved. And we have some other examples of this as well. Teresa showed us a really nice example in the last Myco talk, but it's not always so simple. And so what I'm going to do for the rest of my time is ask in 2 parts, are yeast populations adapting in the human body? And so in the first part, I'm going to tell you a sort of very narrow story about recurrent vulvovaginal candidiasis, looking at just where I live in Manitoba. And then the second part, I'm going to scale up to the world. So vulvovaginal candidiasis, this is what we call the yeast infection. And so this is the one that gets called yeast. It is really, really common. Thought is that about 3/4 of people with vaginas will have a vaginal yeast infection at some point in their life. Most of them are uncomplicated and they're cleared once you take drugs. And what I mean by cleared is that the symptoms go away. And so whether the yeast are completely gone from the vagina or they remain but no longer trigger the immune system, I think is still somewhat up for debate and may differ based on different people. But some appreciable proportion of those people, about 8 to 10%, will unfortunately have recurrent symptomatic cycles. And so this is where 3 to 4 times a year at minimum, they have those same symptoms that come back again and again and again. This is a really, I would say, awful chronic condition. It leads to poor mental health. It can lead to damaged relationships. It can lead to disengagement from medical care. And about 50% of cases are idiopathic. So we don't know why certain people are susceptible to these recurrent symptomatic cycles when so many other people are, one and done or, very sporadic. And so as an evolutionary biologist, this is evolution in action in the human body. This is sort of the same population evolving essentially evolving over time. And so when my lab basically first started, we wanted to ask the question, are the vaginal yeast populations adapting? Can we pick up a signature of adaptation? Or is these other forces sort of counteracting selection? And so to do this, we set up a clinical collaboration with Dr. Vanessa Poliquin, who's a fantastic OBGYN clinician scientist in Winnipeg, where I live. A global pandemic is not a great time to set up new clinical collaborations, but we've been really fortunate to have wonderful people locally. And so what we've been able to do is that Vanessa has a specialty yeast clinic once a month. And from her clinic, we've been enrolling people to take swab samples for us. And so today I'm going to tell you about 4 different participants, one with a Necaseomyces glabratus infection, which shows up as pink dots on our chromagar plates, and 3 with Candida albicans infection, which are these green dots. From each of these 4 people, we took 12 vaginal isolates and 12 rectal isolates. And so we're trying to capture the population. We did fairly standard genomics and bioinformatics. So we whole genome sequenced these isolates. We aligned them to the S5E5 genome with BWA-MEM, called variants by adapting the GATK best practices, and created a SNP file. And then from those sort of SNPs, so from the variants within all those isolates, we generated phylogenetic trees with RAXML, and then we can calculate SNP diversity from all those isolates. So to calculate diversity, we're doing this in a way that accounts for missing data, which is really common with the short read data that we're using. So if you picture 4 isolates or 4 haplotypes, in the first way, we're going to just sum the number of differences between each pair of isolates. So shown here in the pink boxes, there'd be 1 difference between haplotype or isolate 1 and 2, and 1 between isolate 1 and 4. We're also going to calculate diversity using this program Pixie, which again accounts for missing data. We're basically going to average the number of differences by the number of comparisons we're making. So this is pi, which is a really standard metric of diversity. So the first thing we can do is look at the relationship of these isolates, the vaginal and rectal ones. And what we saw in all 4 of the participants, ones from both species, is that the vaginal and rectal isolates are completely overlapping. So this, from this way of looking at it, looks like this is the same population. It's not like there's a separation of vaginal and rectal isolates. And this has been shown before with other types of sequencing, like MLST data showing that rectal and vaginal isolates are closely related. But this is the first time, to our knowledge, that someone's used whole genome sequencing. So really gotten at the nitty-gritty of what's different. And that's really interesting, because when we look at the number of SNP differences, we see that within both the rectal population, so comparing rectal isolates, the vaginal populations, comparing vaginal isolates, there's quite a lot of differences. So if you compare any 2 isolates, there's about 500 SNPs that are different between them, which I think was more than we were expecting, given we expect these sort of consistent population crashes. When we compare rectal and vaginal isolates together, we see the same thing. And so it's not like there are certain, it doesn't look like there are certain alleles that are just in the vaginal population and just in the rectal population. These really look like highly overlapping populations. When we look at the level of diversity, as we would expect based on what I just showed you, we see that the level of diversity in the vaginal and rectal populations are about the same. So those square vaginal populations are more or less about the same as the rectal populations, and very similar across our 3 C. albicans populations. These numbers in isolation don't necessarily mean that much, though. And so we combed the literature for other studies that had done population level sampling. We found 2 of them. So if you were here last month, you heard Teresa talk about one of these studies. And then we also found 3 examples, or one other paper that had 3 other examples. So these were 2 studies that looked primarily at healthy oral populations, and then a couple of individuals that also had rectal populations. I'm downsampling our study to just be 3 isolates, because it really matters how many isolates you put into the diversity calculation. But what you can see is that in the most case, our vaginal and rectal populations taken from women with recurrent vaginal yeast infections have the same level of diversity as many of the healthy oral individuals. There are a couple of individuals that have very high levels of diversity, and this was published in that PLOS biology paper. But for the most part, really similar. that's not what I was expecting. I thought that we would have much lower diversity in these recurrent infections where we expect a lot of bottlenecking. On the Nacocemyces glabratus side, we found one paper that had looked at bloodstream isolates. Now we've downsampled to 10, they sampled more isolates. And ours are, I would say, higher than most of the bloodstream isolates, somewhat similar to some of them. And I think we know that bloodstream isolates go through these bottlenecks. And so again, I was expecting them to be really similar to these bloodstream isolates. So I think all of this begs the question is, you know, where there is obviously some mismatch. There's higher levels of diversity than we were expecting in these populations. And so to somewhat answer the question of, is there scope for yeast adaptation to the vaginal environment? Well, selection critically requires genetic diversity. And yes, we definitely have diversity, but adaptation requires selection to overcome drift and migration. And I think we don't necessarily know if that is possible yet. I think we need more comparable studies. So the question is, are the vaginal isolate populations a higher diversity than we were expecting, or is maybe the healthy oral populations a lower diversity than we were expecting? And so I think we need some more comparables. But I think we found really excitingly is that there is significant diversity in these populations, which sort of begs the question, what is happening in between these cycles of drugs? And this is one of the things that we're currently looking at. Okay, so now going from like hyperlocal one population, we're going to expand to the whole planet. And so when Abdurrahman for the last few years has been scouring the NCBI to pull off as many Candida albicans isolates as he could. And so a huge thank you to all the people involved in these many, many papers who deposited their data there. And so from this, he pulled 1,229 isolates. You can see where they fall. So again, don't pay too much attention to the colors right now, but essentially those different colors are different body sites. And this is the data set we're working with. From those 1,229 isolates, we added 77 new whole genome sequenced isolates from Winnipeg. Abdurrahman kept all the isolates that had at least 80% of reads mapped to the A21 reference genome, at least 80% breadth of coverage, and at least 30x mean depth. And so from that, he was able to keep 1,112 isolates and create a new C. albicans phylogeny. So this is what it looks like. It's rooted with C. africana. So that's this group of isolates over here that look like they're very, very low diversity. So they do map well to Candida albicans, but it is, I think we can agree, we could talk about it, a new species or a different species and should be treated as such. And so with this phylogeny, our first question was, how many clades are there? We took the Roper's 182 isolates, and so the outer ring shows you where the Roper's isolates fall into this phylogeny. For each of those defined clades, we look to see where sort of the monophyletic group would sit. And so these pie pieces show you how far the Roper's clade designations get us in this new phylogeny. And I'm going to briefly deviate to give a short history lesson. Where do these clades come from in the first place? So they come from this paper from 2007, which was an MLST-based paper. So they had 1,391 isolates. So it's taken us about 20 years to get the same number of whole genome sequenced isolates, but here we are. And they chose this p-distance of 0.04 because this cutoff separated clusters that contain known examples of isolates in clade 2 and SA, which is now known as 4, as previously determined by DNA fingerprinting with the moderately repetitive probe CA3. On the basis of this cutoff, 17 clades were defined. And so this was somewhat arbitrary and based on old technology, but we wanted to sort of stay true to this because obviously we don't want to sort of throw it into a washing machine and pull out something completely different. And so we thought, okay, let's see if we can use a statistically aware method that doesn't, throw out these decades of what we've been calling clades. We use this tree cluster program, which is statistically based clustering for phylogenetic trees. It has many different algorithms involved. We ran them all, but I'm going to tell you about what we stuck with, which was called max clade. In tree cluster, you have to pick a threshold size, which again, the details I'm happy to talk about if someone's interested, but we looked for areas of parameter space that had a consistent number of clades. That was a reasonable number. So, between 50, 30, and 10 was kind of what we were expecting. And we found this really nice region of space, which I'm calling A. When we map this program's clades back onto the phylogeny, we were delighted to see that it really well matched the Roper's phylogeny for the most part. And so now I've colored the isolates by the supported clades from Max Clade. There are 20, and by and large, the Roper's clades fit perfectly on top of it. There's a few exceptions. So cluster D and cluster B are now sort of nested in other clades, but those were sort of clusters that had small numbers of isolates to begin with. And from the original MLST designations, we've lost 6 of the clades, and I'm going to call clade 13 as C. africana. And so I would love to discuss with people, because I don't think that we should be making this decision, is how do we name these 20 clades? And so just a quick recap, currently there are 11 numbers and 3 letters. So these are the Roper's designations. We have 6 missing clades from the MLST, and then we have 6 new unnamed clades. So again, love to talk about it afterwards. Okay, so what can we learn from this? So when we first map isolation source onto this phylogeny, we see that it is somewhat biased within clades. And so we see that, for example, there's a lot of circulatory system bloodstream isolates over here, which matches these clades, which was really exciting. However, when we also map geography, we see that they are almost perfectly confounding in lots of places. And so we see that these circulatory stream isolates are also really biased towards Asian isolates. We see these large blocks of North American oral isolates. And this is, a common thing that happens in biology. This happens in lots of systems where you sort of have this confounding of geography and species, essentially. And so we're still working on how to sort of tease these things apart. But a few things that I can tell you from this. One is that the environmental isolates are really diverse. So we're able to get some new environmental isolates from Dana Opulente and Chris Hittinger from their survey of sort of American soil and plants. And so if we map those isolates and the food spoilage isolates and these 2 isolates over here that come from bird feces onto the phylogeny, they are spread throughout the phylogeny. This is what other people have seen with smaller studies before, but it was really nice to recover that. And so we're seeing them sort of spread throughout. We also see a signature of local migration. So we sequenced 77 isolates from our local hospital microbiology lab. So this is essentially every Candida albicans isolate that came into the lab in 2012 as the black arrows, in 2018 as the gray arrows. And so this is mapped onto just clade 1, which is where we see a lot of our isolates. And you can see that they are really sitting right beside each other in a lot of cases. And this sort of spans the isolation source. And so we have this signature of local migration. And so these 2 things combined, I would say, are consistent with horizontal transmission and the existence of stable populations outside of the body. And so I think that there's papers certainly that show that there's maternal inheritance, and there's, I'm sure, absolutely maternal inheritance and sexual transmission most likely. But this does, I think, speak to the very high likelihood that we're also seeing Candida albicans be transmitted from person to person locally. And so to sort of tie both of these parts together, my lab is really interested in asking the question of whether there's scope for vaginal adaptation. So shown here in the outer ring, all those pink lines, those are the vaginal isolates. If we zoom in again on clade 1, which is where we see a lot of them, we do see them clustering, somewhat closely together, regardless of the geography. And so we see vaginal isolates from very different countries sitting right beside each other. And so we are currently, again, looking for a genetic signature of vaginal adaptation. One of the problems, though, is that we don't necessarily have the phenotypes to test. And so people have certainly gone looking to see if there's phenotypic differences between vaginal isolates and other isolates. And for the most part, our in vitro tests show absolutely no difference. And so I think it's still an open question of, if there is adaptation, what are they adapting to? What is the predominant selective force? And we're hoping that we can take a genetics first approach to ask that question and then follow it up with confirmation in the lab. And so with, I think, the couple of minutes that I have left, I'm going to a little bit stand on a soapbox. we thought 1,112 isolates was tons. This was really hard work. I didn't at all highlight how difficult it was for Abdurrahman to create this global phylogeny, but it actually turns out it's not sufficient. And so it's pretty biased what we have. We need more South American, Eastern European, Middle East, African, Australian isolates. Australia isn't on this graph because we have somehow no isolates from there. We're missing large swaths of the planet, and I think that we're still missing large swaths of the diversity of the species. We've had a lot of isolates that come from the bloodstream, and we've had now quite a few from the oral, but we're missing lots of other contexts. So I think if people are sitting on isolates from other sites, that would be fantastic. I think untargeted studies are really useful to help us answer these questions. And I will also pitch that we could really use more intra-population sampling in different contexts so we can put our diversity metrics into context. We don't actually yet know whether the numbers that we've recovered are high or low because we just don't have the baseline. We don't know what the baseline is. And so with that, I would like to say that there are many forces acting on yeast populations. Adaptation requires selection to overcome drift and migration. Genetic diversity is sufficiently high for selection to act within populations and at the species level, but migration is also prevalent within populations and at the species level. And so I think a lot of us are interested in all kinds of different disease contexts. And I think that, ultimately evolution, I'm biased, I'm an evolutionary biologist, but, nothing in life can make sense. Nothing in life can make sense except in the light of evolution. I really do think is true. And I think that with using sort of this framework or sort of this viewpoint, we can better understand how these species are causing disease if we put them in the context of what's happening throughout the body and throughout the planet. And so with that, I would like to first and foremost thank the participants of our clinical sampling, which we call Thrive. We can't get isolates without participants, and we're really grateful to the people who are willing to sample for us. Our clinical collaborators have been fantastic. So Vanessa Poliquin, Alicia Berard, and Fren Mulhall, again, starting a clinical program coming from where I've come from is a really difficult thing to do. And Alicia runs studies on bacterial vaginosis. Fran was a nurse working in the lab at the time, and they were both instrumental in helping us get this off the ground. Diagnostic Services Manitoba, which has been fantastic in giving us access to the isolates that come into the microbiology lab. And all the funding sources I've had throughout the years. And I also just really want to quickly say thank you to MOMI folks and the MBL. When I started my postdoc, I had never touched a human fungal pathogen in my life. And through that course, I gained the confidence to do so. And I'm delighted to say I think I've now have started or published on every single species that we worked with in the MOMI course. And so with that, I'd like to say thank you again for this opportunity. And I hope for some great discussion.
Well, thank you very much, Aleeza, for such an exciting and very thought-provoking talk, and I'm sure there will be lots of discussion going forward. So just to remind everyone that we won't take questions now. We will take questions at the end of the meeting. And I see some people are putting their hands up in the participants. Please remember, questions are taken through the Q&A function at the bottom of your screen. So please do use that function if you wish to ask questions, and it would be very good if you can indicate to which speaker those questions are targeted at. Okay, so it's my very great pleasure to introduce my friend and colleague, Professor Stuart Levitz from the University of Massachusetts Chan Medical School. So in addition to being a really outstanding scientist, Stu is a clinician, and he consults on infectious diseases at UMass Memorial Hospital as well. So I consider Stu to be one of the founding members of modern medical mycology. He's been working in this field for over 42 years. He's I had a look in PubMed, and his earliest paper that I could find was from 1983. And he has been publishing prolifically since then and still continues to publish. He's got over 200 publications, all in the areas of medical mycology, focusing on fungal immunology and vaccinology. And his current active area of research is in the development of vaccines to protect individuals at risk from Cryptococcus, which we'll hear about shortly. Now, those of you who have heard Stu speak before know he has a very good and dry sense of humor, which no doubt will come out shortly. And in his very brief and modest bio that he sent over to me for this introduction, he ended that with that he loves to discuss mycology with anyone who will listen to him. I'm sure then that he's delighted that he's got now a captive audience of 130 people for the next 30 minutes. Stu, over to you.
Oh, thank you, Gordon. Appreciate the kind invitation, and let me and the kind introduction. I anticipated the kind introduction with this slide over here, and I have to say, like Aleeza said, the MycoTalks have been great, and particularly during the pandemic when we were sort of isolated as a community to be able to hear everybody's science. So it's a great honor to be able to talk here today. As most of this audience knows, fungi have really gone viral. David Denning's Lancet ID paper estimated 6.5 million invasive fungal infections a year, with almost 4 million deaths, 2.5 million attributable to fungal infections. The numbers may be a little inaccurate because fungi aren't, for the most part, reportable diseases, but regardless, a lot of people are getting sick and are dying from fungal infections, which is why when I started, there weren't that many people studying it. And now, fortunately, there are a lot of people studying it that are doing great research. So what are the drivers of this perfect storm that are causing these 6.5 million fungal infections a year? Mostly immunocompromised. We live in an age of immunocompromise with AIDS, transplantation, chemotherapy, immunosuppressive drugs, etc. Those are putting our patients at risk. Medical devices, antibiotics, climate change is expanding population, is expanding fungal habitats, infection control really difficult as fungi are everywhere. Antifungal drugs have a limited armamentarium and resistance. And what I'm going to talk to you today about is, and hopefully working on rectifying the problem, is that there are no fungal vaccines. So there's a lot of societal obstacles to fungal vaccine development. A lot of the fungal infections are in developing parts of the world, and getting the vaccines to developing countries and the expense of clinical trials, not only in developing countries, but everywhere, are tremendous. Public and MD acceptance of vaccines seems to be going down, especially from the leadership of some countries. Unfortunately, mine also, although I will say that vaccines remain very, very popular, and over 90% of people do get vaccines, at least childhood vaccines. And then there's a legal climate, which some companies are reluctant to get into vaccine trials because of that. There's also scientific obstacles to vaccine development. Persons most at risk for fungal infections are often immunocompromised and don't respond well to vaccines. Also, fungi and humans are closely related, and this limits the number of unique antigens which are available. And, here we are, and here we are, I guess. At least I'm not a fungus. I don't think so. And here are the fungi. So really a branch of the eukaryotic tree over there. And, here are plants which are pretty far away from us. So despite all of these obstacles, 3 fungal vaccines have actually been tested in humans, although none is FDA approved. It does show that it is possible to do human testing for fungal vaccines and even human trials. There's a formula in spherules from Cox City coccidioides, which unfortunately was very reactogenic, and the study was stopped early without an efficacy signal. There was a pan-fungal subunit vaccine with beta-1,3-glucan conjugated to a protein carrier that was tested in humans but never reached clinical trial. And there's the vaccine from Jack Edwards' group with Scott Filler and Ash Abraham and others with the ALS3 protein vaccine, which was safe and immunogenic, and there was a trend toward fewer episodes of vulvovaginal candidiasis, although none are in clinical use right now or FDA approved. So our research is focused on developing vaccines to protect people at risk from cryptococcosis. And with Jenny Lodge and Raj Upadhyay, we're studying a vaccine that lacks the three chitin deacetylase genes. It's a whole organism attenuated vaccine, either used live or heat killed. And Jenny discussed this during her MycoTalks a year and a half ago. And I'm not going to talk much more about this vaccine. Rather, in the limited time that I have, I'm going to talk about our work with subunit vaccines. So just to give you a little bit of background on Cryptococcus, and there was a nice article from David Mayer and Peter Williamson in the New England Journal of Medicine recently, but Cryptococcus is an encapsulated fungus. It's the only medically important fungus that has a true polysaccharide capsule. There's a strong association with disorders of T-cell immunity, especially CD4 T-cell immunity. So particularly those with AIDS and are on T-cell immunosuppressive medicines, such as people with transplants. And meningitis is the most common clinical manifestation. So who could benefit from a Cryptococcus vaccine? Probably mostly people with HIV and AIDS. There's approximately 150,000 deaths a year. Importantly, over half of these cases are in persons that have been on antiretroviral therapy in the past. So these are people that have been identified as being HIV positive. They've been put on HIV medicines. So there's an opportunity when they're identified to vaccinate. And it's estimated that 19% of AIDS deaths are due to cryptococcosis. Solid organ transplants, people get vaccinated. If you're put on an organ transplant list, you're generally vaccinated prior to your transplant when you're not immunosuppressed or not severely immunosuppressed. And there's an approximately 0.5 to 4% lifetime incidence of cryptococcosis. Endemic regions, Cryptococcus gattii, the sister species of Cryptococcus neoformans, is endemic in tropical and subtropical regions, Vancouver Island, the Pacific Northwest, and this region seems to be expanding somewhat, perhaps due to climate change. And I also want to mention that koalas, sinonasal infections are common due to the association of Cryptococcus gattii with eucalyptus trees. And as with developing chlamydia vaccines, which koalas get a lot of chlamydia, I think there should be more of a focus on looking at Cryptococcus vaccines in koalas. So our four-step approach to a subunit cryptococcal vaccine, identify and recombinantly express protective CD4 T-cell antigens, define surface antigens on Cryptococcus that elicit protective antibody responses, combine the antigens with adjuvants that elicit strong antibody and Th1 memory T-cell responses, and then vaccinate and protect the at-risk population from cryptococcosis. So I'm gonna, since I never do anything in the right order, I'm gonna start out by talking about step 3, which is combine antigens with adjuvants that elicit strong antibody and Th1 memory T cell responses. And the adjuvants we found out early were really, really important because when we started out working, most of the adjuvants that were available, and especially FDA-approved adjuvants such as alum, they stimulate very good antibody responses, but not very good T cell responses. So our vaccines mostly gave modest protection, if any protection at all. And looking at it retrospectively, the reason was that they weren't stimulating very good Th1 or interferon producing CD4 T cells. So we started out looking at glucan particles, and for this, this was the brainchild of Gary Ostroff, close collaborator and friend at UMass. And what Gary did was he figured out a way of, he took baker's yeast and he chemically treated it to leave one with a beta-1,3 glucan shell that was porous. And then he figured out a variety of ingenious techniques to load antigens or proteins onto the glucan particles. And when we took antigens, cryptococcal antigens that were loaded onto glucan particles, and then we immunized mice with these glucan particle antigens, what happens is that the mice gets fooled into thinking that it's under fungal attack, and it's sort of a fake news to the immune system, and you get the type of immune response that you see during a fungal attack. You have a strong antibody response, and you also have a Th1 and a Th17 response to the antigen. And so then from there, we went to identify and express protective CD4 T cell antigens. And we had a variety of methods that was sort of biased, unbiased, agnostic, whatever you want to call it. But by these methods, we came up with a bunch of different antigens that we tested. We cloned them, expressed them in E. coli, loaded them into glucan particles, and tested them for protection. And we've tested over 24 recombinant antigens now in both BALB/c mice and Black 6 mice. We've also used mice with humanized MHC2 epitopes, DR4 mice. And this is somewhat of a complicated slide, and it summarizes at least our first 24 antigens. And each black dot represents a mouse that's been vaccinated, and the day that the mouse died. So let's say for this vaccine with this protein, the mouse died on day 32 over here. And you can see, and the red vertical bars show median survival. So this, for the BALB/c mouse, for the CD8-2 antigen, these mice survived the entire experiment up until day 84. And you can see different antigens protected differently, and there were differences between the BALB/c and the Black 6 mice, and that's shown here also for the humanized DR4 mice. In the absence of vaccination or with vaccination with an irrelevant antigen, the mice all die before day 30. You can see here for the BALB/c and the Black 6. And based on these data, the data with the humanized mice and other factors, some of which I'll get into later, we're moving forward with these 5 antigens as our lead antigens. So we were excited by these data, and I would talk to vaccine companies, big pharma, NGOs, startup companies about the glucan particle vaccines and how great they were and how amazing the protection was. And basically, I would get a thumbs down. They would say it's very interesting. Glucan particles may work great as an adjuvant, but they may be too expensive to formulate, especially for use in resource-poor countries. And they've also never been tested in humans as a vaccine adjuvant, so they would need separate testing, and there would be concerns about that they could be very reactogenic. So I was at a Gordon conference a bunch of years ago, and I ran into Ash Ibrahim, and we were sort of discussing adjuvants, and Ash recommended trying CAF-01, cationic adjuvant formulation 01, which is made by Staten Serum Institute, a nonprofit in Copenhagen. And it's a liposomal Minkel agonist, which elicits antibody and Th1, Th17 type T cell responses. So, this is sort of a schematic of it. It has a synthetic version of mycobacterial cord factor. And like Dectin-1, Minkel stimulates through Syk card9 pathways to lead to inflammatory responses, which includes Th1, Th17 type responses and antibody responses. So we tried that, and it worked similarly, maybe not quite as good as glucan particles, but almost as good. And these are individual vaccines, so single subunit vaccines adjuvanted in in CAF-01. And all of them showed variable degrees of protection in BALP-C and especially in Black 6 mice. So if one was good, maybe more would be better, and indeed more was better. So for BALP-C mice, if we did a 2-antigen combination, we saw 70% protection, and it went up to 100% protection with when we used 4 antigens. For the Black 6 mice, 2 antigens protected 100%, and then we lost protection we didn't have the Minkel knockout mice, but when we used the card9 knockout mice, which is an adaptive protein for Minkel, we lost all vaccine-mediated protection. And we then looked at clinical strains. The work that we had done was with a strain KN99, which is a highly virulent strain of Cryptococcus, but we were interested in clinical strains from AIDS patients, and we tested 6 different clinical strains. I know this is sort of a complicated slide, but the dashed lines show protection, vaccine-mediated protection. Solid line shows mortality of the mice in the absence of vaccination, and the vaccine protected against all the different strains, although the protection was somewhat variable depending on the strain. So Gordon is probably asking, what's the immune mechanism of protection, being the fungal immunologist that he is? So first of all, CD4 T cells are required for protection, regardless of which vaccine that we use. if we take mice that lack CD4 T cells or not shown on the slide, if we delete CD4 T cells with a monoclonal antibody, protection is completely lost. If we do similar experiments with mice that lack CD8 T cells, or if we deplete CD8 T cells with a monoclonal antibody, we do not lose protection. So this shows that the CD8 T cells are important and the CD4 T cells are important, and the CD8 T cells seem to be play a minimal role in protection. We've then gone on to look at the phenotype of the CD4 T cells recruited to the lungs following vaccination and infection. We've looked at memory T cells in unpublished data, and also at cytokine skewing and data that has been published. So for the memory T cells, and I know there's some controversy in terms of which markers are used for memory T cells and different T cell subpopulations in the lungs. This is the schematic that we did, and our gating strategy. And when we looked at CD4 T cell subsets, what we did was we looked at unvaccinated mice and mice vaccinated with our 4 antigen regimen in adjuvanted and CAF01. This is day 0, so this is prior to infection. This is day 10 of infection, and this is day 70 in the survivors. The unvaccinated mice don't survive to day 70. And you can see that by day 10, there's in the vaccinated mice, there's a robust response of T cells into the lungs, and the T cells are predominantly effector memory and resident memory T cells. By day 70, the lungs have mostly recovered, but there are still some memory T cells in the lungs. Similarly, recall lung CD4 T cell responses following vaccination and infection. So this is unvaccinated, uninfected, vaccinated, uninfected, unvaccinated, infected, and vaccinated and infected. And you can see you have a nice CD4 response in the lungs. These are activated T cells. Looking at the activation marker CD154, these are Th1 T cells. So these are the interferon producing T cells. These are IL-17 producing T cells. And these are CD8 T cells that are making interferon gamma. You can see you get a little bit of a Th1 response, but not much of a response. And these are recall responses to some of the vaccine antigens. So if CD4 cells are necessary for protection against Cryptococcus, will vaccines protect patients with impaired CD4 T cell function, such as those with HIV? And, this is a question that I don't have a experimental answer to. I will say that you can vaccinate at-risk persons early in infection, such as pre-transplant or in HIV when the CD4 count is still high. There's a lot of evidence for Cryptococcus that a lot of disease is activation of a latent pool of cryptococcal infection. But the other way of doing it is to try to elicit protective antibodies. So you figure the patients don't have good T cell responses, but if you can elicit good antibody responses, you can overcome the lack of T cell responses. So this is our step 2, defining surface antigens. And when we did this in mice, these are with glucan particles. We have similar results with CAF01. But what we found was with our two antigens, looking at CDA1 and CDA2, there was no phenotype in mice that were pretty much lacked antibody, suggesting lacked B cells and antibody, made very little antibody. And this suggested that antibody wasn't playing a role. But as in probably some of your dating profiles with immunology, immunology is always complicated too. And if we look at two of our other vaccine antigens, now we saw a nice phenotype in the mice that were vaccinated but lacked B cells and had very little antibody response. So what's going on here? How's antibody protecting? It could be neutralizing a virulence factor. It could be opsonizing. There could be growth inhibition. And a clue came from a paper from Peter Williamson's group, where he isolated capsular proteins from DMSO extracted capsule. And 3 proteins, LHC1, BLIP4, and CPD1, were associated with the Cryptococcus capsule. And coincidentally, these 3 proteins are 3 of our lead vaccine antigens, or maybe not coincidentally. So we immunized mice with these 3 antigens, and we took serum from immunized mice, and by immunofluorescence, you can see that antibody nicely coats the surface of the capsule, sort of in a punctate pattern, which makes you think that perhaps this is vesicles. And you can see a monoclonal antibody that reacts with glucuronoxylomanan, the capsular polysaccharide, gives you more of a diffuse type pattern. So this showed that there was antibody on the surface. And then we also showed that you can get opsonophagocytosis with vaccine induced antibody. And the amount of opsonophagocytosis was similar in mice vaccinated with, I'm forgetting which antigen this was now. I think it was BLIP4 compared with the monoclonal antibody over here. So moving into humans, which candidate antigens also protect humans and perhaps zombies? And for this, we teamed with a postdoc, Jessica Hartigan, and Peter Williamson's lab, and Peter, and we got frozen PBMCs that were, and then we both from patients with cryptococcosis without known immunocompromise, and also from healthy donors. And then we either left them unstimulated, we stimulated them with the super antigen SEB or tetanus toxoid as positive controls, and then heat-killed Cryptococcus or recombinant Cryptococcus proteins. And then we measured a stimulation index after looking at activation antigens on the T cells. And you can see that all of the cells, regardless of whether they were from healthy donors, and each color is a different donor. The healthy donors are in circles, and the crypto patients are in triangles. And they responded about equally to mitogens and to tetanus toxoid, showing that they didn't have an intrinsic defect in their T cells. And then if we looked at heat-killed Cryptococcus, the patients responded better than the PBMCs to stimulation with the heat-killed whole organisms. You know, what about the vaccine antigens? A little or perhaps a lot less robust. There were trends toward greater responses in the crypto subjects. This was significant with BLIP4, but compared with the healthy donors. So what does this all mean? I think given the diversity of MHC alleles in the population, you likely need multiple antigens in a vaccine. And also the mouse data suggests that protection requires immune responses at the time of initial infection, which is an additional argument for vaccination. So in terms of step 4, you know, this is still aspirational. We're hoping to partner with a startup. We are planning on picking our top 3 antigens, maybe synthesize a chimeric protein. We have decided on CAF01 as our adjuvant, and you know, and then if we can get through these 3, hopefully get to phase 1 trials to test for immunogenicity. So in conclusion, recombinant antigens that are adjuvanted with either glucan particles or CAF01, they protect mice from Cryptococcus. Protection varies as a function of mouse strain and also cryptococcal strain and species. CD4 cells are required. Antibody may contribute to protection against selective antigens. Cryptococcal antigens stimulate CD4 T cell responses in human patient PBMCs, and we're poised to move toward human testing of a CAF01 adjuvanted subunit vaccine. And if any of the 100 and so participants listening want to fund this or partner with us, please get in touch with me. Most important slide, of course, is the people who did the work. I want to particularly thank my longtime partner in crime here, Charlie Speck, and also the other members of the lab, Maureen, Lorena, Susie, Satya, Diana, Zach, some of the lab alumni, the collaborators who I've mostly mentioned already, support from the NIH, which at least as of today hasn't been cut off, and also my medical school. So thank you for listening.
Thank you very much, Stu, for a really nice overview of the vaccine and the work you've been doing over the last few years. And what I forgot to mention is that you've been funded continuously by the NIH, so hopefully that will continue in the current climate. All right, if Aleeza, if you want to join us on screen, there we go, and I will hand over to Sarah, who will kick off the questioning.
Great, thank you both so much. Just a reminder to the audience, please keep submitting questions through the Q&A, not the chat. And we will try to take them, and Gordon and I will handle them. So we have several questions for Aleeza that are coming in. So there were questions tending to relationship of when the patients were taking drugs relative to when samples were collected, and maybe you can speak to that.
Yeah, so the easy answer for that is that they had to have been off antifungals and antibiotics for the past at least 2 weeks before the samples were taken.
Okay, and I guess sort of as a follow-up, are you planning any studies where you're going to try and do that in a more prospective manner?
Yes, so in addition to this sort of snapshot in time, we now have 30 people with a history of recurrent vaginal candidiasis who've taken samples for us for a month, so 3 times a week. We have multiple sites that we have some samples from. And so we're getting both yeast isolates when the population size is high enough. We're also intending to do, the full microbiome workup of both the fungal and the bacterial. And I'm submitting a big grant on Tuesday, which we would love to do like 6 months of sampling, because within that month, we're getting a big diversity. Some people are coming in with high yeast populations. Some people are flat the whole way through. And so we'd love to sort of get a bigger breadth of people to capture symptoms and populations as they cycle throughout a longer period of time.
Yeah, so I assume your expectation in that is that you're going to have reduced diversity as you treat with drug. But given what you found so far, what, if you had to make a bet, what do you think the outcome's going to be?
Yeah, so we have a few people. So again, this is like there's one special yeast clinic once a month. And so naively, we're like, people will come in with high yeast populations, they'll be prescribed drugs, they'll take the drugs, the population will crash. Turns out you don't get to pick when you get that appointment. And so for the small number of people we have who have come in high yeast, take drug, population crashes. In some cases, although we don't recover yeast on our plates, we still see that if we look at like ITS sequences, there's still Candida albicans in the microbiome. And so, you know, 0 on a plate doesn't necessarily mean 0 in the population. Surely the population is crashing in terms of population size, but I don't think it's going to 0. I think that there's still a population that's being maintained there. And it seems like, you know, there's room for a lot of migration coming in from other body sites. And so the diversity might also be maintained sort of at other body sites and then can come in. Yeah.
Gordon, do you want to take over?
Yes, thanks, Sarah. So Stu, a couple of questions that are sort of vaguely related in one way, but not in the other. But anyway, the first one is about antigenic drifts. So if you target a vaccine and a fungus, will it evolve away from that specific antigen? Will there be antigenic drift away from protection?
I don't think so, but I can't say for sure. There is data from Arturo and other groups that Cryptococcus micro evolves, but I don't think there would be enough selection pressure to cause it to micro evolve. One of the selection criteria we did use for our vaccine antigens was that there was a high degree of homology between Cryptococcus neoformans and Cryptococcus gattii, so that and also across a lot of the genomic database. So I don't think that there would be that much selection pressure in a human, but it would remain to be seen.
Okay, well, your answer has made me skip the second part of my question, because there was a question about whether you tested your cross-reactivity against Ketia in your experimental models.
Yeah, we did. With our glucan particle vaccines, we have published some work with gattii, and in general, it's more difficult. The gattii strain that we've used, the only one that we've tested has been the R265 strain, which is gibberish to most of the people listening, but it's a hypervirulent Cryptococcus gattii strain, and it's been more difficult to protect against the gattii strain compared with some of the neoforman strains that we have looked at. So, but I don't think it's because of antigenic differences. I think it's because of differences in Cryptococcus gattii, and maybe gattii being a more virulent organism, but it is a problem that we need to work on more.
Okay, and then coming to my second part of the question, which is related, but from the host side, really. Talking about the differences between your bulb C and C57 mice, you saw differences in protection, right? There's probably
Yes.
differences in MHC. So the question was whether is that what you think it is in terms of epitope loading, or
Right.
and did you use perhaps make F1 mice to see whether you could see an impact of that, and how is that impacting your idea to treat humans with different HLA?
Yeah, we've been talking for a long time about doing F1 mice, but we haven't done that. So, but what we have done to look at the allelic differences in MHC class 2 is we've compared C57 black 6 mice with what are known as DR4 mice. So DR4 mice have the MHC class 2 alleles deleted and replaced with the humanized DRB04, 01, whatever allele. So the only differences between the DR4 and the black 6 mice are in the MHC class 2 alleles, and we do find differences in protection with some of our antigens when we compare vaccinated black 6 mice with vaccinated DR4 mice, and we've gone on to map the epitopes. Some of the epitopes are similar, so they seem to be what we call promiscuous epitopes, but most of the epitopes are different between the DR4 and black 6 mice. And therefore, when you think of a T cell vaccine and trying to protect the general population, and there's over 10,000 different MHC class 2 alleles that have been described. Some of them are minor differences, but some of them are more major differences. That's why I think you probably need to have, if you're talking about a subunit T cell vaccine, I think you need probably need multiple proteins in order to see protection. And you need to define proteins that have epitopes that react with the alleles that are seen in the general population. And in addition, when you start thinking globally, the alleles that are found in certain parts of the world, I see in your background, you have what looks to be the continent of Africa. Alleles that are seen in parts of Africa are different than alleles that are seen in Exeter and in the United States.
Okay, thanks. Over to you, Sarah.
So Aleeza, we have a question about whether I'll just read the question. That these similar levels of diversity that you see in your different fungal populations, whether this is a byproduct of a requirement for multiple genes, and that therefore this diversity is in itself evolutionarily maintained?
Yeah, that's what I've been thinking about. So kind of getting back to this idea that there's so many selective pressures acting on these populations that you kind of need to maintain sufficient diversity to deal with all of them. And so there's often sort of trade-offs between evolving to one thing versus evolving to another. So you could imagine that evolving, to be a specialist at pH4 might be really bad for dealing with fluconazole. I don't know if that's true, but you could imagine lots of scenarios where that would be true. And so in some case, I almost wonder if selection is acting to maintain the alleles that let you stay as a generalist species rather than as a specialist, and which means that you're not getting strong selection for any one allele to sweep through the population. And so at any one time, you have, lots of things kind of interacting to keep that diversity and to stop the organism from becoming a specialist.
And so is that, broadly speaking, what would define the evolution of a commensal compared to a pathogen?
I mean, I think so. And this is what I'm coming around to, is that
now that we're in the era of sequencing where we can really do this for all kinds of organisms.
Yeah, I think that's a really interesting idea. And I've been thinking a lot, like what are other species where we see certain things like this? The hard part is that a lot of the theory has been developed with recombination in mind. And so, right, where you have sex and all your genomes are mixed. And so all these things like linkage disequilibrium and ways that we can statistically sort of get around this migration versus selection balance don't hold for asexual or primarily eusexual microbes. And so also thinking about what those models look like when you primarily or only have clonal reproduction. I think there's a lot of work from some mathematicians out there.
Yeah. There was a question, I don't know that this can really be answered, but whether the isolates you were identifying, can you really classify them as commensals versus pathogens?
I mean, I think lots of people have looked for like, what does that mean, right? So we have like a gut morphology, maybe we have some morphologies that are maybe more common as a commensal or I don't know that we can define an isolate. I think that most isolates are capable of both growing happily without stimulating the immune system and also capable of growing and stimulating the immune system, which I think is what we define as pathogenic. And so I guess, I've been thinking a lot even about the words we use. So I try not to even say recurrent infections. What does that mean, recurrent infection, in this scenario where you probably have some level of saying genetic variation all the time. So what we have is recurrent symptoms, right? And so I think we also have some like linguistic issues, which is everyone's favorite part of science.
Yeah. Oh, right. No, absolutely. Well, the words we use matter a lot. So I had a question about whether you see if you, because obviously all your VVC isolates are coming from women, but is there any change in diversity, or can you cluster when you look at your oral or systemic isolates, whether there's any difference between those coming from men and women, particularly premenopausal? Because I'm thinking, you know, we know estrogen is, of course, one of the number one risk factors for having VVC, and whether another issue might be the sex of the person carrying it.
That's really interesting. We haven't tried to stratify anything by sex in our like diagnostic services Manitoba collection. We obviously have both men and women and a whole big diversity of ages. For our VVC studies, it's like 18 to 50 premenopausal only. And we would love to expand this to like people with neovaginas and postmenopausal people who also do sometimes get yeast infections, but
your oral, right? Presumably the oral
yeah,
have roughly, and if there's any clustering of the samples from women.
we haven't looked. We have lots of information about the participants on our RVVC side. They fill out extensive questionnaires. We have less information from the microbiology lab samples, but I haven't thought about clustering by sex. That's a good idea.
All right, I'll turn it to Stu. And Stu, can I ask Stu a question? I want to know about koala T cell biology.
Sounds like it sounds like you're ready for a second sabbatical in Australia, and I'll go with you too. I think we're all.
Australian roots, you know.
I don't know anything about koala or about koala immunology. I do know that there's a lot of work looking in with chlamydia because koalas get a lot of chlamydia, and I think people are studying ways of uh, of treating the chlamydia and vaccines against chlamydia. I don't know anybody who's looking at vaccines against Cryptococcus. There are some studies that about 5 or 10% of koalas in the wild have cryptococcal antigen in their blood, and you can find colonization in their sinuses, and some of them actually do go on to develop sinus disease, and it's more common in koalas in captivity. So it's a problem for wildlife parks and zoos, etc. So there is interest in it, but I don't know, and I presume they have Th17 cells, but I couldn't swear to it.
We're interested in Th17 for sure. But let's get back to humans now and medical mycology. So there's a couple more questions here. First of all, one's related, I guess, to Aleeza's talk about the diversity of fungi in populations and whether that would impact your ability to develop a vaccine. Stu.
Yeah, that's really interesting. I saw that question. I mean, Stu, you could talk too. So things like Nakasaomyces glabratus, which I didn't really talk about, but if you look at that, we're building a whole genome or a whole species tree for that. I mean, that really looks like a species complex. I think by the metrics we usually use to delineate species, there are really big differences between what we currently put under the umbrella of Candida glabrata or Nakasaomyces glabratus.
So the antigens then, Stu, do you see variation between different strains?
Well, the antigens that we're looking at and some of the part of our selection criteria was that there was high homology between Cryptococcus gattii and Cryptococcus neoformans. So in general, there's, it's up in the upper 90% in terms of the homology, and the antigens that we're looking at are very conserved. But I think, you know, the answer to the question in terms of vaccines is, yeah, you want to have targets that are present in pretty much all of your strains or almost all of your strains of and species of fungi that you're trying to target. And you know, if not, you're going to miss too many of them.
Okay. And then the last question that was asked online was around mRNA vaccine technology and whether you're exploring moving into that.
You know, it's funny you mention it because just this month, Zhirong Lin in Georgia published a paper in NPJ Vaccines, and she looked at two of our vaccine antigens, BLIP4 and CDA1, and she made RNA vaccines, and neither one protected mice by themselves. And then she went on and she added an adjuvant. She had this capsular adjuvant. And then with the adjuvant, the mice were protected with one of the vaccines. And I think the problem with RNA vaccines is they don't really give you robust CD4 T cell responses. So if one is going to use RNA vaccines, you probably need to have a good adjuvant with the RNA vaccines so that you can at the same time stimulate good Th1 responses. Th1 responses are really, you don't get it if you don't have a good Th1 response, it's going to be very difficult to get, I think, good protection against Cryptococcus.
All right, thanks, Stu. Aleeza, I have a question for you. Not understanding really fungal genetics very much, but what you showed, as I understood it, was that when you looked at specific patients and you looked at the candidate patients, there was a very narrow distribution, but you said you were surprised that there were 500 SNPs between the different sites, right? You showed those very nice bell-shaped curves. So I was wondering whether you looked at the identity of the genes behind those SNPs and whether those SNPs, you always see them in the same between the different candidates, and also if you look at that between, say, bloodstream isolates from the same patient, do you see that? Is there a correlation in this?
This is exactly what my second PhD student is working on for part of his thesis, is to look to see if we can see gene reuse in different patients and also different species. So if we see the same genes emerging and as being mutated in vaginal isolates across different patient populations and across different species, that would be a huge indication that those are the genes that are involved in adapting to the vaginal environment. And also this is sort of like a genes first approach to try to get at what the phenotypes might be, because if we know something about the genes, maybe we can then guess what they're adapting to in the vaginal environment, rather than saying a priori, let's look at growth in our PMI, or let's look at, you know, biofilm formation. There's been a bunch of papers that have looked at in vitro phenotypes that don't find an association, so we'll take the genes first approach.
Okay. And then I have a last question for Stu, and this is my question, Stu. So the problem with vaccinating people who's immune compromised, you pointed that out in your animal models and of course in patients.
Yes.
You don't have T cells anymore, and that's not going to work.
Yeah.
But I was wondering whether you'd ever tried an approach where you immunize an immunocompetent animal and then deplete the T cells and see whether there's protection, because presumably if you vaccinated an HIV-infected individual, there'll be enough B cell activity still going on in those individuals that could lead to protection. Is that a possibility?
Yeah. So can you see the screen I'm sharing here? So
Okay.
what we did was I knew somebody would ask this question, so I had it ready here. So what we did was we vaccinated the mice, and then we depleted the T cells prior to Cryptococcus infection. And if the T cells it's sort of a complicated slide here, but we did see vaccine mediated protection, and it was lost as the T cell depletion increased. So if you have partial T cell depletion, you still see protection. If you have complete T cell depletion, you lose protection. So and that's one of the reasons. And with the vaccines that we use, they're not good ones on the surface of Cryptococcus. So maybe, maybe with some of the antigens and with combination antigens, we would do better. But it's certainly a problem. And I think if you have no T cells, you're exposed to Cryptococcus, the vaccine probably is not going to be very much effective.
Okay. Thanks very much.
All right. Well, thank you to both our speakers. We had one request from the audience to remind everybody listening that the MOMI course run by University of Chicago at Woods Hole, applications are still open. They're open till March 17th, and encourage people to sign up. I'd like to thank both of our speakers for incredible talks and a fun discussion. Thanks to the audience for chiming in. And thanks as always to the University of Exeter MRC Center for supporting this, and all the people behind the scenes, Jenny, Spencer, especially for making this run as smoothly as it does. All the glitches that you did not see are because of her. Anyway, thank you all, and we'll see you next month.
Thank you, everyone. Goodbye.
Yeah, thank you.